Light is created when electrons within the atoms become excited by absorbing extra energy from their surroundings. The extra energy causes the electron to move up to a higher energy level within the orbital shells of the atom. Since atoms seek stability, the electron will return to its original orbit, releasing a particle, or photon of energy in the process. A ray of light is a steady stream of photons traveling along in an electromagnetic wave. The amplitude is the height of the wave and is the distance from the dashed line to the wave's crest or trough. The amplitude squared is proportional to the intensity. Wavelength, the distance between two crests, is the characteristic of a light wave that defines what type of light it is, X-ray, visible, or ultraviolet. A light wave can travel in any angular plane perpendicular to its directional axis from 0 to 360 degrees. A light wave is made of two components, an electric and a magnetic. These two components travel in sinusoidal waves along the light path at right angles to each other. The wavelength of light within the range of visible light determines the color of the light wave. White light is made up of many rays of varying wavelength. The speed of light remains constant in a vacuum, but wavelength and frequency change along the electromagnetic spectrum. Light waves with shorter wavelengths have a higher frequency and higher energy. The speed of light is constant in a vacuum at 3 × 108 meters per second. The speed of light outside a vacuum depends on the refractive index of the material it is passing through. When a beam of light passes from one material to another, it changes speed, causing the light to bend. This is called refraction. The refractive index of a material is typically greater for short wavelengths, so light waves with shorter wavelengths such as violet bend at a sharper angle than those with longer wavelengths such as red. As a result, white light traveling through a prism is split into a rainbow. A convex lens will act as a magnifier. When the object being viewed is inside the focal point, a larger virtual image is formed behind it. While early microscopists used simple convex lenses to magnify objects, there were obstacles to overcome, such as chromatic and spherical aberrations. While a convex lens will refract white light as a whole through its center, light passing through the edge of the lens will have different focal points for blue and red light. This is called chromatic aberration. Spherical aberrations result when lenses used in microscope objectives send rays of light into a sample at different angles of incidence, depending on the distance from the center of the lens. Because the angle of refraction at the cover slip will be different with increasing distance from the center of the lens, the rays of light will come to a focal point at slightly different depths within the specimen. The virtual image reflected by a plane mirror will appear to be as far behind the mirror as the real object is in front of it. It will also appear at an equidistant angle from the mirror as the real object. Modified reflective mirrors are used in incident-like microscopes for imaging opaque specimens. If a single light wave were split equally in two, they would produce two light waves with half of the intensity of the original beam. In the first box, we see that if these two light waves remained in phase, so that their crests and troughs were aligned, the sum of the intensity of the two light waves would equal the original wave. In the second box, we see two light waves exactly out of phase, mixing to cancel each other out. Box 3 shows light waves that are slightly out of phase. In Young's experiment, light is completely blocked by a plane, with two pinholes comparable in size to the wavelength of the light. Light waves passing through the pinhole will spread out in all directions. When light rays from the two pinholes hit a screen and are in phase, a bright spot appears on the screen. When the rays are out of phase, a dark spot is seen. The property of diffraction limits the microscope's ability to differentiate between individual particles on a slide. If two structures are close together, their diffraction patterns will interfere with each other, and they will be seen as one structure. If the two structures are far enough apart, their diffraction patterns will overlap in such a way that they can be distinguished as two separate structures. A microscope's ability to distinguish between two structures is known as resolution. The theoretical resolution limit of a far-field optical microscope is approximately half the wavelength of its light when using an objective with a numerical aperture of 1. The discovery of the law of refraction in 1621 made possible the development of the compound microscope. The basic compound microscope consisted of two convex lenses in series, an objective lens close to the specimen, and an eyepiece lens near the observer's eye. Light microscopes may illuminate the sample from the opposite side of the lenses, known as transmitted light, or illuminate the sample from the same side as the image-forming lenses, known as reflected light. Modern research labs often use inverted microscopes for materials and cell cultures. The microscopy suite of the Imaging Technology Group has a Zeiss Axiovert 200 light microscope equipped with a variety of filters, a fluorescent lamp, motorized stage, and digital camera linked to a computer. Like most modern microscopes, this scope is inverted. The parts of the microscope will be presented in their order along the light pathway. The Axiovert 200 uses a tungsten lamp for a transmitted light source. The condenser is a lens system which gathers light from the source and concentrates it to a cone of light that illuminates the specimen with uniform intensity across the field of view. The condenser must be configured properly for each objective. A research-grade microscope may have a special turret for holding multiple condensers which can rapidly be exchanged depending on the imaging technique being used. Techniques such as Nomarski DIC, phase contrast, and dark field illumination all require special condensers. A computer-controlled motorized stage allows the user to move the specimen with fine adjustment as well as collect multiple tiled images over time. Probably the most critical component of the optical microscope is the objective lens. The basic function of the objective lens is to gather light passing through, being reflected, or being emitted by the specimen, and then to project an accurate, real, and inverted image of the specimen into the body of the microscope. The objective can also house special devices for contrast methods, such as an iris for dark field microscopy, phase plates for phase contrast microscopy, or correction collars for different cover slip thicknesses. The Photometrics CoolSnap is a computer-controlled digital camera embedded in the scope. The second image-forming lens system in a compound microscope is the eyepieces, or ocular lenses. Eyepieces work synergistically with the microscope objectives to further magnify the intermediate image so that specimen details can be viewed with the human eye. Different types of eyepieces exist, and usually the eyepieces are matched to the types of objectives mounted on a microscope base. Selection, position, and focus of the objective, condenser, and diaphragm can be achieved via the computer or manually using the scope controls on the right side of the instrument. The fluorescent lamp control box is a separate unit attached to the scope. This is the stage control box that allows the computer to manipulate the stage. The resolution of an optimally designed objective is dependent on a number of factors. 1. The refractive index of the material through which light rays pass between the sample and the microscope objective. 2. The cone angle of the light collected by the objective. And 3. The wavelength of the light used for imaging. The refractive index, or N, is defined as the relative speed at which light moves through a material with respect to its speed in a vacuum. Here, N equals 1. Air has a refractive index of 1.00029. Light rays traveling from the cover slip of the slide with a high N will change speed and diffract when they pass into air with a low N. A 10x objective with an NA of 0.25 is capable of gathering enough light rays from a distance to form a clear image magnified 10 times. But for a sharp image at a higher magnification, better resolution is required. This can be achieved by a 40x objective with a cone angle of 31.3 degrees, resulting in an NA of 0.52. For a higher magnification of 100x, better resolution is needed. Since light rays with a cone angle greater than 72.1 degrees will not pass from a high-index material like glass to a low-index material like air, the largest numerical aperture for an objective with air between the objective and cover slip is 0.95. When oil with an index of refraction of 1.52 is placed between the objective and cover slip, the numerical aperture can be raised to 1.45, increasing the resolution of the objective. Kohler illumination is a method to evenly and adequately light the area of the specimen to be viewed. The microscope can be aligned by first making sure that the lamp has warmed up sufficiently to get the maximum amount of voltage from it. Next, adjust the light so that it is positioned directly in line with the condenser and diaphragm. Using low magnification, move the stage to the area of the specimen you are interested in and zoom into that area. Next, align the condenser by adjusting the height and lateral position to center and focus the aperture image. Then open the diaphragm to fill the viewport with a lighted image. Finish aligning the condenser by fine-tuning the position of the diaphragm and condenser to adjust the contrast of the image. Contrast in biological tissue sections can be enhanced by staining the sample. The most common stain is commonly known as H&E and is made from hematoxylin and eosin. Hematoxylin will bind to nucleic acid and will highlight the cell nucleus. Eosin binds to the cell cytoplasm. After the sample has been stained, transmitted light is used to image the sample. A light wave is made of two components, an electric and a magnetic. These two components travel in sinusoidal waves along the light path at right angles to each other. A light wave can travel along its primary axis at any rotational angle. The electric vector can be arbitrarily oriented relative to the microscope. When a polarizer is added to the light path, the orientation of the electric and magnetic vectors are defined. A polarizing filter is composed of long-chain molecules arranged in parallel rows. Light waves oriented parallel to the molecular chains are absorbed or blocked by the filter. Light waves oriented perpendicular to the molecules are not blocked by the filter. Many geological samples are anisotropic, meaning that light rays with perpendicular polarizations are absorbed differently. For example, one polarization may be absorbed more strongly than another polarization, or a specific color may be absorbed more strongly in one polarization than another. White light, which is composed of varying wavelengths, travels from its source at different polarization angles and varying intensities. The beam passes through the microscope's polarizing filter, resulting in light waves that are plane polarized. Since the optical properties of the sample are dependent on the orientation of that sample, rotation of the sample results in striking changes in the color and intensity of the individual crystals. An analyzer, or second polarizing filter, is used to select the polarization of the light to be sampled at the eyepieces or camera. In transmitted light microscopy, light rays pass through or around a specimen. Dark-field illumination is a contrast method in which the central light rays are blocked out using an opaque annulus, and only oblique rays are used to illuminate the specimen. This method is useful for highlighting outlines, edges, and boundaries in a specimen. Simple dark-field systems can be fashioned by placing an opaque disc at the center of the cylinder of light entering the condenser lens. If no specimen is present, the entire field of view will appear dark. When a specimen is present, only light which has been refracted or reflected by interacting with an optical feature in the specimen reaches the image plane. Because the illumination is oblique, uninterrupted light rays will pass outside the entrance to the objective lens. Dark field microscopy creates images that appear as brightly illuminated structures on a dark background. Reinberg illumination is very similar to dark field, with the addition of contrasting colored filters used for the oblique and transmitted light rays. This results in a brightly colored image on a contrasting background. If a single light wave were split equally in two, they would produce two light waves with half of the intensity of the original beam. In the first box, we see that if these two light waves remained in phase, so that their crests and troughs were aligned, the sum of the intensity of the two light waves would equal the original wave. In the second box, we see two light waves exactly out of phase, mixing to cancel each other out. Box 3 shows light waves that are slightly out of phase. In phase contrast microscopy, a ring of light illuminates the sample. When these light rays interact with the sample, some are bent, resulting in changes in path length for the light rays and phase retardation. A phase ring is used to change the phase of the unbent rays, maximizing the contrast that results for mixing the out-of-phase rays. Positive phase contrast is achieved by delaying the diffracted waves by 1/4 wavelength. This produces a dark image on a light background. Reflected light microscopy is used for opaque specimens. Differential interference contrast, or DIC, is often used with reflected light microscopy, as well as transmitted light microscopy, to enhance edges, making the image look more three-dimensional. This is accomplished by adding a polarizer, a Wollaston prism, and an analyzer into their appropriate places in a light microscope. The Wollaston prism works by splitting each ray of light into two parallel rays. In a reflective light microscope, shown here, each ray strikes the surface of the specimen and is reflected back at different angles, depending on the optical properties of the specimen at that location. The prism recombines the split rays, now slightly out of phase. The interference pattern created by the two waves produces varying degrees of contrast in the final image. The contrast of the image can be enhanced and fine-tuned by moving the prism and adjusting the analyzer. Fluorescence microscopy is a common method of visualizing the structure of a cell. The specimen can either autofluoresce or be treated with a chemical capable of fluorescing, called a fluorochrome. When a fluorochrome molecule is exposed to a particular wavelength of ultraviolet or visible light, usually in the blue or green wavelength range, its outer electrons absorb the light and become excited, jumping to a higher orbit. When the electrons return to their ground state, there is a loss in energy, resulting in the emission of a photon with a longer wavelength. The wavelength range that a fluorochrome will absorb and the color it will emit is dependent on the chemical structure of the fluorochrome. Microscopists will use different fluorochromes to tag antibodies to various parts of the cell, so that the nucleus, for example, will fluoresce a different color than its surrounding cytoskeleton. There are many different methods used in fluorescent microscopy. The following describes how an autofluorescing substance is imaged. White light passes through an excitation filter, which allows only light of a specific wavelength range to pass through, while all other light is blocked. The light beam that passes through the filter, blue in our example, is reflected by a dichromatic mirror through the objective and onto the specimen. If the specimen fluoresces, the emission light gathered by the objective passes back through the dichromatic mirror. The combined emitted red light and excess reflected blue light pass through a series of barrier filters, which will remove the blue light. The remaining red light represents the fluorescent areas of the specimen.